Can we imagine a virus so deadly it kills every single organism it infects? This isn't a scene from a sci-fi horror movie, but a grave reality. A reality born in a lab where doctors trained by the Chinese military decided to play God with nature, the creation, an engineered version of the pangolin coronavirus, loosed upon an unsuspecting population of mice. The results were staggering and terrifying. Every single mouse infected by this man-made monstrosity died, an unyielding mortality rate of 100%. The disease was swift and brutal. Within a mere 5 days, the infected mice lost significant weight, became lethargic, their eyes turned an eerie white. And by the 8th day, the life had been cruelly snuffed out of each one of them. But the virus didn't stop there. It invaded more than just the bodily functions. It went for the brain, the eyes, the lungs. A rapid, unexpected spread that left the mice's internal systems ravaged and their bodies empty shells. The virus's deadly spike in brain infection was particularly surprising to the scientists. A chilling discovery that left them with a haunting question. Could this virus potentially infect humans? All the mice, without exception, succumbed to this engineered nightmare. The scientists were shocked by the results of their experiments. But what does this spell for us, humans? The experimenters were taken aback by the devastating effects of the virus they had created. The mice, once healthy and full of life, were reduced to lethargic creatures within days, their eyes turning a haunting white. Their weight dwindled rapidly, and in just over a week they were gone. A grim end indeed. But the surprises didn't stop there. Post-mortem examinations unveiled an alarming discovery. The virus had not just attacked the mice's respiratory systems, as one might expect from a coronavirus. It had infiltrated their eyes and even their brains. In the final 2 days, the brain infection spiked dramatically, a shocking revelation that left the scientists reeling. This brought forth an unsettling question. Could this virus, tailor-made for mice, pose a threat to humans? The scientists themselves warned of such a possibility. They suggested that severe brain infection, which proved fatal in mice, might be a key cause of death in humans too, should they contract the virus. So why was this virus created in the first place? To better understand the potential risks, to prepare for them, and hopefully to prevent them. But the path forward is filled with uncertainty and trepidation. The virus, initially designed for mice, may well be a lethal threat to humans. The question remains, why create such a deadly virus in the first place? In a world grappling with the realities of viral pandemics, it's a question that echoes in the minds of many. The paper published on the open access website BioRxiv offers some insight, but it also raises more questions. According to the researchers, the creation of the pangolin coronavirus was a part of an experiment to understand its effects. Yet the lethal outcome for the infected mice and the surprising spread of the virus to their brains, eyes, and lungs was unexpected. The scientists were reportedly surprised by the results, stating, "This finding suggested that severe brain infection during the later stages of infection may be the key cause of death in these mice." However, the high pathogenicity of the virus, particularly its ability to cause total mortality, demands further investigation. The paper stated, "The high pathogenicity mechanism of GX_P2VC7 in HACE2 mice, in the absence of the wild-type GX_P2V control, requires further investigation." In simple terms, the scientists need to understand why this virus is so deadly. They need to explore the specific mutations in the virus that might be contributing to its lethal nature. They also plan to conduct further tests, but with a crucial difference. Instead of using the lab-created virus, they intend to use the actual virus found in nature. This shift in approach could provide valuable insights into the behavior of the virus in real-world conditions, rather than in a controlled laboratory environment. This research is not just about satisfying scientific curiosity, it's about understanding the potential risks and consequences of such viruses. It's about preparing for the worst-case scenarios and developing strategies to prevent or mitigate them. As we peer into the abyss of this potential catastrophe, it is clear that understanding these man-made viruses is not just about scientific curiosity, but a matter of survival.